Thank you all for coming. Thanks to the folks in Zoom land. I'm Bob Sandler, and it's my distinct pleasure to introduce our visiting professor. This is going to be a longer introduction than usual, but this is the first time we've had a vice chancellor speak at our research conference, so bear with me. So our visiting professor is John Carethers, and John was born in Detroit. He was the 10th of 12 children. He attended college at Wayne State in Detroit, where he graduated with a degree in molecular biology and biophysics. He then attended medical school at Wayne State, in part so he could live at home, and he graduated at the top of his class. He was an intern in internal medicine at Mass General Hospital, where he was one of only 3 of 23 interns who had attended a public medical school. He was recruited to be a GI fellow at the University of Michigan by the legendary Tachi Amada. And this is an example of how stuff happens perhaps by accident. So while he was a fellow, he was mentored by Rick Boland, who at that time was studying misratch repair. There's a whole story behind that I won't go into. But when John completed his fellowship, Rick Boland had gone to UC San Diego, and he recruited John to become to follow him there. When John got to San Diego, he assumed positions of increasing leadership. He was first the director of the fellowship program. Then he was chief of the GI section at the VA and eventually chief of the GI division, a position that was opened up when Rick Boland moved to Dallas. So during John's tenure as chief, he doubled the size of the faculty, he doubled the size of the research portfolio, and he obtained a center grant similar to the one that funds CGIVD. In 2009, John returned to Michigan as a chair of the Department of Medicine at the University of Michigan, where he served for 13 years. While at Michigan, he oversaw a $400 million budget, 930 faculty members, and $250 million in annual external research. And then in a bit of symmetry, he was named the Richard Boland Professor of Medicine, a chair that was endowed in honor of his former mentor. On January 1 of this year, John was named the Vice Chancellor for Health Sciences at UC San Diego, and he replaced our former Division Chief David Brenner in that position. John has many honors. I'll tell you a couple. So he was elected to the American Society of Clinical Investigation, the National Academy of Medicine, the American Academy for Arts and Sciences. He's a fellow of the Royal College of Physicians. Scientifically, he's published about 200 papers on various aspects, largely of colon cancer. He's been an invited speaker all over the world. He's mentored about 100 trainees, many of whom are now leaders. Last year, Barbara Young was a visiting professor. She is the incoming president of the AGA. She worked in John's lab. So on a personal note, I met John about 20 years ago, and this is another example of how stuff happens by accident. So we were at a meeting and we were chatting, and I said, you know, I got all these bloods and tissue specimens from colon cancer patients, and he needed that stuff for his research because he didn't have a local resource. So we've written about 6 publications together, and we continue to send his lab specimens. As recently as 2020, there was an abstract that was published. We also served together on the National Commission of Digestive Diseases, and I got to serve with John on the AGA governing board. The title of his talk is Environmental Influences on Colorectal Cancer. Thank you for coming.
Thank you so much, Bob. It's been a pleasure seeing you in your element here. And thank you for having me. You know, we're going through that discussion of my career. A lot of things happen in happenstance, and I really thank Bob for many years of partnership and publications that we've had as well. It's great to be here. The weather is similar to La Jolla today, so that's really good. Which I've been there for about 7 weeks. And I will see David Brenner next week. Again, I've already talked to him a couple of times in preparation for taking this current role that I have. So I moved there about 7 weeks ago, and my lab is still at Michigan. I intend to move it this spring and continue some of the work that we've done for a while. I hope most of you are at DDW this year. It's May 5th through 9th. Please be there. We're looking for your registration, your presence, your science, and our engagement and networking. So my title today is Environmental Influence of Colorectal Cancer. I have, let's see, make sure I can move this. Is that moving? Yes. These are my disclosures, just NIH and formerly University of Michigan, now UC San Diego, a little bit of research funding. I'm an independent director on Avantor, but there's no relation to what I'm going to talk about today at all. And I'm on the scientific advisory board for a private company called Geneoscopy, and there's going to be no discussion of anything with that as well. So this is the outline for my talk today. So I'm just going to go through some general concepts of genetic environmental concepts for colorectal cancer. Then I want to give you an example, particularly some of the work I do and others have done, where genetics change the environment of the tumor, and then how the environment can change the genetics of the tumor. And then just have a little closing discussion on the microbiome environment as we learn things. Now, some of these things I hope I give some new concepts, but if not, you can throw a brick at me if you wish. Since this is the world of GI, everyone who, particularly in the clinical world GI, but in the research world, knows there has been this phenotypic change that occurs as we develop cancer in people. And typically, as you grossly look at this, it starts out as a small polyp grows, and then some percent of those polyps can acquire other molecular features that cause it to invade and perhaps kill the patient. These are just some endoscopic pictures of those. Many of you have seen these. Come in a wide variety of looks for that variation. When you think about cancer, about 1/3 is actually familial, based on a family history. 2/3 are sporadic. And even of the familial, we don't understand the monolithic forms of inherited diseases for a small percent, includes Lynch and FAP, and some of the hamartomatous polyposis syndromes. 2/3 are sporadic. What we have time studying some of these rare syndromes, many of the things that we've learned apply to the sporadic such that that information could also help address the approach to their treatment therapy. And Jeff, if there's any fellows in the room, in the next slide are good slides to have for your. This is just the list of the major genetic forms of the anomalous polyposis syndromes, where it's typically one gene or one allele that causes those syndromes. They list the chromosome location. They shouldn't test you on that. Test you on if it's recessive or dominant. That's all common, since I wrote some questions in it. And then some of the even rare adenomas like MSH3 associated polyposis, which is described in the last 5 years. The next slide is the hamartomatous polyposis syndromes, which are even more rare. The understanding the genetics has led to grouping. Now we grouped several as P10 hamartomatous polyposis syndromes because we know it's the same genes that cause some of these phenotypic variants. For instance, like juvenile polyposis, where you can get ING and SMAD4 and BM mutations. So one question is, what is the biggest driver? In those genetic diseases, the genetic are the biggest driver. You get a mutation, you get a variation from anywhere from 20 to 100% certainty that you may have cancer. Now, is that true for sporadic cancers? Talk about these gene-environment interactions. This was done a little over 20 years ago. It was twin studies done on cancer, published in the New England. Say it's around 50,000 twins, of which 10,000 had cancers. And what this looked at, they used the mathematical model, taking monozygotic, i.e. identical twins, and dizygotic, i.e. fraternal twins, and look at the contribution of shared gene in non-shared environment, shared environmental, non-shared environment, and potential hereditary, their mathematical model. Now, the very interesting thing about this paper was that 20 cancers they looked at, they said, based on their mathematical model, only 3 were primarily driven by a large genetic component. The first one was prostate cancer. 28%, I believe, of what went into a prostate cancer, 42% what went into a prostate cancer was genetically driven. The next highest was colon cancer, 35%, and then breast at 28%. And all the other ones dropped off statistically based on this model. When you look at since those were genetic components, everything else based on was driven by environment. So particularly for colorectal cancer, 35% drives the genetic, and 65% is the environment, of which 60% was non-shared. And so they concluded that all principal and dominant role in causing sporadic cancer based on their model. And if you think about this, we're approaching 8 billion people in the world. We're all genetically different. We have, subtle SNP differences, subtle polymorphisms, but on your genetic makeup of your 25-ish thousand genes, you can be susceptible to things in the environment that vary from the next differently susceptible person. You're exposed to something in an environment, that could be anything in the air to something you use recreationally or whatever, and in combination give you your susceptibility to cancer. The way I think of it is versus non-modifiable factors. What are the things that you change versus things you cannot? Are some of the things what do you carry in your germline? A high risk monolith like an APC mutation, or a low risk compendium of alleles that give you a risk based on the particular makeup. And collectively we call that the family history, as best we can tell. There's your racial ethnic, because there's differences based on that background, and we just call that background genetic makeup. The other thing that you cannot change is your age. Someone has a time machine, okay? On the ones, these are the things I know Bob has studied for many years, and many of you are studying. What you ingest, how does that change the exposure? Use of tobacco or hormone replacement therapy. And the one thing that the physicians in a room can change physicians is the screening utilization. So that's a variable component. And of course, the biggest push that is probably the easiest thing amongst you to change, particularly in the United States or worldwide, is getting screening utilization up. It's a great equalizer. And I can give you a whole nother talk. So think about non-modifiable versus modifiable. So the non-modifiable are typically the genetic things, maybe the age. Modifiable is the environment. Now, when you have, the environment can do a lot of things to a cell. And this is just an example. I do work in the area of DNA mismatch repair, so it's repair mechanisms. but a lot of things can happen from the environment to alter that repair. So we're bathed in oxidative damage, particularly in the colon. So much the UV light in the colon, but certainly on the skin and other things. We have modification of many things that we eat, so everything from xenobiotics to other things that are modified by metabolism, as well as other organisms that our gut has. We can have spontaneous deamination of sinus slippage of DNA repair. And all those things can contribute to damage. They can alter transcription, they can alter protein expression, and they could be mutated, happen in, particularly in that can drive an early polyp or cancer. Now, in thinking about the sporadic cancer, which is the bigger portion of that, this is the Cancer Genome Atlas network analysis that summarizes about 35 years, about a decade ago, but it still stands as work. And so I'm just this briefly. On the upper graph, notice the y-axis is the mutation rates. These are somatic mutations found in the cancer. And then notice it's a log scale. And on the x-axis is various descriptions of the type of genotype of the tumor. The MSI status, the CpG island methylated phenotype status, and the MLH1 silencing. They're all referring to a failure of repair by inactivating one of the mismatch repair genes, MLH1. And notice that the approximately 15% of all sporadic colon tumors have 1,000 mutations occurring, and they are all caused by a defect in mismatch repair. There's a handful of what we call hypermutated tumors. This group of 15% is outlaid in this inset box. And the ones that are really hypermutated, green dots on polymerase. So it's a mutation in one of the places that can give you 1,000 to 2,000 mutations, whereas the mutations are of one of the mismatch repair proteins, MLH1, can give you a hypermutated tumor. The other 85%, anywhere from 3 to maybe 8 mutations that drive that tumor. So these are two different complete pathways, and even though both of them can give you colon cancer, they actually are two complete diseases. And if you look at the genetic downstream effects from these two groups, the ones that are hypermutated, where you take out mismatch repair, notice somewhere between 25–50% of genes are taken out, and for the most part, it's by the gene because they have coding microsatellites, and when you take repair, you slip at those microsatellites. Most of them are non-coding regions. The ones that are coding are. This is in contrast to the 85%, which I'll call another name, Vogelgram, named after Bert Vogelstein. Chromosome instability, few mutations, and the mutations are very common in all of those tumors, APC, P53, KRAS, etc. And then you get this long tail that are by as an individual fingerprint to a tumor that is vastly different from person to person. The majority are those 3 or 4 tumors. So this is 35 years of work summarized in just a couple slides. Now, so this is, I just wanted to present that concept before I started getting into the genetics. The other thing I'll mention, and this also, again, that's genetics and environment, is this work by Andy Chan on the Mass Journal from the Nursing Health Professional Study and the Health Professional Study, in which he took people people with colon cancer, and keep in mind, who have a primary colon cancer have a higher risk for second colon cancer. This is why we screen them more often. But he assayed whether the first they had, did it have a mutant PI3 kinase, one of those genes you saw in the Vogel. And it turns out that if you have a mutant PI3 kinase and you took aspirin to have the benefit of aspirin to suppress the occurrence of a second tumor than if you had a wild type. So this is where genotype can predict how the environment will affect. So looking at this graph here in panels A, C are patients who had a mutant PI3 kinase in their primary tumor. And you can see that those who didn't take aspirin had a higher probability of death compared to those who had used over a 10-year period. And this was true for colorectal cancer specific mortality and overall mortality. Type PI3 kinase, excuse me, in your primary tumor, aspirin did nothing for you to suppress. So these things you approach to personalized care thereafter if you know some of the genetics. So the way I think of this is you can have genetics that drive FAP or Lynch, in which different aspects of tumor progression are a genetic defect, or you can have sporadic where these things happen over time, but then each one of those stages can be influenced by the environment. And I have here microbiome and inflammation as influencers at any one of those stages. This is even true if you have a genetically driven thing. So FAP or Lynch, you can have 3 of someone with an LH1 mutation, but the grandmother developed her cancer without any intervention at 40. Mother may not develop one, but the kid at 28 developed cancer. And that's modified, even though they're 70 to 80% risk, it's modified by other things such that the expression of that risk can vary even with the same genetic mutation. The other constant, the level of inflammation and immunity on these tumors. It goes back to some of the work that Jeremy Galland did out of Europe, and he's published probably another 100 papers since this paper. Looking at the type and density of immune cells in these tumors and how it can affect outcome. And so we tumors stage 1, 2, 4, where one, survival rates are 95+%. In stage 4, maybe it's less than 10%. But if you the CD3 sites, which maps all lymphocytes, essentially, and look at the center of the tumor and the invasive margin of the tumor, or IM, if they were high, suggesting there was some immune relationship to the activity of the tumor, irrespective of their stage, you can predict a much improved survival compared to those who had low tumor center and invasive margin lymphocytes. And of course, those with already stage 4 who had metastases were already had poor disease-free survival. I want to mention those concepts, the meat of this talk. So I want to get of how the genetics actually change the environment. In other words, how do genes modify such that the environment is different? And the example I'm going to use is DNA mismatch repair. So just as an evolutionary conserved DNA repair mechanism that goes all the way down to bacteria. This affects every cell in your body. It's most active after DNA is replicated to ensure the fidelity of replication of the DNA. In humans, these repair proteins work as heterodimers. They work together. So for instance, MSH2 also works with MSH6. MSH2 will also work with MSH3. Are slightly different. 2,6 will repair single nucleotide mispairs, and 2,3 repair longer deletion loops. I call this the broken zipper model. And so you ever taken a zipper and it somehow got squicked and then one piece sticks up and then it's hard, your zipper gets stuck. How many times has that happened to you? Well, this repair system is that extra loop. On this example shows that this AG, a tetranucleotide TTC on the opposite strand. But it doesn't matter which AAG pairs. And if it doesn't pair exactly right, if there's 9, it should pair with 9. But if 8 of them pair, there's a loop out of 4 nucleotides. So that's called an insertion loop or deletion loop, depending on the strand of DNA, the template strand, synthesized strand that occurs. The system will recognize that. Drop the exonuclease and you resynthesize and put the number of repeats in there. And if it can't do that, it may signal for the cell. Now, in most somatic cells, that's fine. This could happen in every somatic that you're shedding in your colon, but who cares as long as it's stem cells, right? So what are some common features of cancer with defective mismatch repair? So that in hyper-MLH1, I showed you up to 15% of all colorectal cancers have a mismatch. And it tends to happen in older patients, okay? The tissues display something we call microsatellite stability, which is those repeats, just the changes. So we call those frame shifts. And that you can easily do. At most academic centers now, every colon cancer is tested for the staining of those mismatched proteins. And so you will see MLH loss from the staining of the tumor, and you will see PMS2 loss because of the instability of PMS2 not binding to MLH1. You have to differentiate, was it at a sporadic for that? But these 70% are located in the right colon. You guys all know this. Differentiate, about 40% have mucinous components, more so than a normal. They're hypermutated, as I showed you in that slide from the cancer. Unlike the Vogelgram tumors, which are chromosome breakages and losses, these tumors are diploid, and so they lack a lot of p53 mutations, unlike the Vogelgram. Now, the thing, how does this change environment? Turns out we have 1,000 microlite sequences throughout our gene. There's only 500 of that 100,000 in a coding sequence. Most of these are not in non-coding sequence. Those approximately 500 genes, when they frame shift, make the full protein. They make a truncated protein, which your immune system has never seen before. That induces those T cells that you see around these tumors, and they form subcellular nodules, and we call these neoantigens. And that anti-PD-1 works for these tumors. So it's the genetics, the frame shift, actually drive the immune response to that. I could give you a whole nother talk on the 5-FU resistance, so I'm not going to talk about, but these tumors are more 5-FU resistant, a longer survival compared to someone who doesn't have MSI high tumor. So just a little more on those lymphocytes. This Vitali back in the thousands, 2008, I believe, yes. What he did is he extracted the T cells from the tumor itself, and you could predict when they had these microcell sequences in the coding, you can predict where the truncation would be because predict where the shift is. And he took cells and he showed that they had immune activity to these novel antigens. But if you took someone who had a microcell stable tumor, no activity. Now, the fascinating thing about this was he took sporadic as well as Lynch MSI high patients, because all Lynch high, and the T cells responded. He also took the T cells from the peripheral blood and shows that these are circulating, so these are longer living, they can go throughout the person's body. So an MSS patient with CR with colorectal cancer had a response, and fascinating, someone who had a germline mutation already identified it as a Lynch, but never had a cancer, already had a response, an immune response. This means essentially self-vaccinating themselves already. And go back to some of the early data from when mismatch repair was discovered. You can see, it's the janitor, he's going to clean the room. But you can see tiny amounts of these frame shifts on even before the peripheral blood. It's probably enough to stimulate the immune system. It's like getting your first COVID shot and you booster. Okay, the patients who are Lynch syndrome, they're already self-immunizing against these neoantigens, which are made by these tumors. Okay, this is also Jeremy Gold. All these turn into memory T cells. So if you take someone who has an MSS tumor and you look at the maturation of lymphocytes, you get very low level. But those with MSI high tumors, because of the education of these needs, you produce a much higher immune response, a memory immune response, showing that this is really immunizing the patient. So the concept here is you can take mismatch repair by a number of mechanisms, sporadic or Lynch, and then you go through the cancer, but you inactivate genes and their subsequent proteins, immunogenic, that stimulates the immune system, and then that stimulates L1, such that checkpoint inhibitor therapy is wonderful for this. And so the concept is you have alternation of mismatch repair, you develop these targeted mutations, you induce a protective T cell inflammation, and the logical consequences are reduced metastasis. Think of this as are like you're Alcatraz, you know, who can escape Alcatraz, right? And you're the wall or the parts, okay? Now, I'm not going to talk about 5FU, but in the studies we've done with 5FU, the tumor cells are much more resistant. So when you give 5FU to a person with an MSI tumor, although no one's done the large study, some of the immune cells. And so some of these people with MSI tumors actually they don't do better, and some of them do. But tried true, you know, so the right study has to be done. So I talked about the immune checkpoint inhibition. So several groups, particularly the former Hopkins now Cornell group, has done studies in which if you have a mismatch repair deficient tumor, most of these in the blue, you see the tumor marker load drops with anti-PD-1. The radiologic response shows shrinkage of these tumors, such that if you have the repair, you had 78% response compared to 11% response with mismatch repair. So these mismatch repair tumors, these immunogenic peptides, are the ones responsible, much more responsive to immune checkpoint inhibition. And thinking about this, so you can have germline or somatic inactivation, you develop hyperimmunity, mutated DNA, get frame shifts at these microsatellites, about 500 are actually encoded. So the important ones to change the environment where you've got immune checkpoint susceptibility. Okay, so you have decent survival, but immune checkpoint can improve their survival compared to a assess. So that's an example where the genetics actually change the environment. So what about environment changing? So I'm going to go back to mismatch repair and just to touch base with this again. So this is just a diagram I drew looking at the main mismatch repair proteins. Arrows show their heterodimers. So MLH1 pairs with the PMS2, but MSH2 compare with MSH6 or 3. Okay, and if you of the type of microsatellite repair, okay, this gives you how TI has defined what MSI high, the old term of MSI low, and this term that's relatively EMAS. So MSI high defined by several white paper mononucleotide and dinucleotide repeats, frame shifts. MSI low was almost all exclusive dinucleotide. Use that term as much anymore because now we know a lot more about EMAS as we're continuing to learn more. And I'll touch base on that in a second. EMAS is under the almost pure portfolio of MSH3. Okay. So what is EMAS? Satellite alterations that selected tetranucleotide repeats. And that was the term. I didn't invent the term. This is more common than classic mismatch repair. This is observed in 50% of all sporadic cancers plus other cancers. I was talking to someone earlier today about ulcerative colitis. The tissues displayed as EMAS. This frame shift mutation at these longer repeats, like I showed you in that one diagram where MSH3 pairs with MSH2 and you got the broken. The tumors are distributed throughout the colon. They are not hypermutated when there's three defect there. So they're not getting a lot of mutations there. If the portfolio of MSH3 is dinucleotide or longer repeats, it turns out that most immunogenic peptides, they have coding mononucleotides, not di. So the number of genes with mononucleotide, probably about 400. With di, maybe 50 to 100. You start getting tri, which are mostly tetranucleotides. There's only 1 or 2 genes in the human genome that has that in the coding. So you don't get all these wild frame shifts like you would do if you take out some of the major proteins in mismatch repair. They're also associated with inflammatory cells, and that was to what was going on. The cancers, we did a nice study showing that they're still sensitive to 5F. Fortunately, unlike classic MSI or Lynch, if you have EMAS, your outcome is much poorer. And we've shown in studies it's higher amongst groups like African Americans who have a higher and poorer outcome from colon cancers. So this is a study, and since Dr. Sandler mentioned Rick Boland, this study was done by the Boland Group in 2012, outlining it. This was a study of, I think, it was about 150 patients or so. If you had MSI high in your tumor, great. But if you had this EMAS, you had the worst survival of all. And if you had neither, you were in. This was statistically significant and was as statistically significant as stage. Okay, so So having this was a poor prognostic. Now, the problem with this, so why did it take so long to figure out this was MSH? And MSH3 is probably the most understudied amongst these DNA misreplicated proteins. Well, it turns out that, so once this was recognized and once it was understood of MSH3 was, everyone was looking for mutations, but no one could find mutations. So we knew MLH, when it goes on sporadic, it gets hypermethylated. It really doesn't get mutated, it gets hypermethylated, so it doesn't get expressed. In Lynch, you have a mutation in one of those mismatched repair genes. But when he had this EMAS portfolio, no one could find a mutation. So what the heck was going on? Okay, so the portfolio suggested MSH3, no one can find a genetic mutation. So some of the clues came, was the hint that came from the association with inflammation. So in the classic MSI, I showed you that mutations cause the immune cells to come in. So we couldn't prove that because the portfolio and the coding microscope stage 3. But we did see some other clues, and those immune cells, many of them produce cytokines, right? And so this is a study showing that these EMAS positive ones had a higher proportional with 6 a pro-inflammatory cytokine compared to those with EMAS negative. So we were wondering, well, you know, could the immune cells be doing to see this EMAS portfolio? And that's exactly what we found. And by the way, we looked at several other pro-inflammatory cytokines and showed it was really just the driver. So this is the key picture from that particular paper. So this is looking at cancer cells. The green staining stains for MSH, blue DAPI stains for the nucleus, and then the third picture is the overlay. And you can see as the green overlaps with the blue, and it tells you that all MSH3 is in the nucleus of these cells. The green, this was a surprise to us. This was an accidental discovery compared to the blue, and the green went outside the blue, meaning that the MSH3 somehow exited the nucleus, which was a shocking surprise to us. And if you look at this by doing nuclear cytosol separation, this is like separating the egg yolk from the egg whites, and seeing and following the proteins, the same thing happened. So you can give increasing doses of IL-6, and if you look at the nuclear western for MSH3, it dropped precipitously by giving IL-6 and was picked up in the cytosol, just switched over. But this was not true of the other DNA repair proteins. MSH2 didn't move, MLH1 didn't move. And histone, as you know, are nuclear proteins, so it tells you the purity, and tubulin tells you the purity. The other thing is, so with that, you give IL-6, then what happens? So we knew that frameshift mutation not repairing. So this is an intrinsic tetranucleotide site, MYCL1. And you can see in the control cell, very low mutation, but the ones that got IL-6, the mutation was 8 to 10-fold. And this was just observing cells after just a couple of weeks of there. So this is a total shocking surprise that inflammation can now inactivate DNA mismatch repair by a completely different mechanism, by translocating a repair protein from the nucleus to the cytosol. So that model says, okay, maybe there's some initiation of neoplasia and tumor advancement. Maybe the microbiome is involved. Trigger inflammation, and you alter repair function, so it's MSH3, so you lose the nuclear function because it shifts to the cytosol. And this is a very common finding, completely understudied in colon cancer. So one thing I also didn't tell you, and I can give a whole nother talk on this, on research, that remember the portfolio dinucleotide are longer frame shifts, but they are also involved. There's several papers now showing it's involved in homologous recombination repair. So this works in the BRCA1 BRCA, and you can get double strand break or loss of double strand break repair with MSH3 defects. So it's double hit to some of these tumors. But we do know that biologic consequences are poor outcome in largely. Well, there's a lot of work trying to figure out why that is. So again, already that's the classic mismatch repair coding. But in this model, you have the environment, the inflammation. So however that is triggered, causing a defect in a specific mismatch repair gene. And then you can get potentially long frame shifts and double strand break and have a completely different outcome than classic mismatch repair outcome. So that was a total surprise to us, and I'm sure I can actively tracking down in that field. So the last part I want to talk about, it's got to be triggering, at least in that last example, something's got to be triggering those things. Now, we don't have 100% proof. We've done a lot of work with some of our microbiome folks on cold culture and certain microbial bacteria. Some of them I want to talk about now, and look at what happens to but it's not quite ready for prime time yet. But that interaction, what else? For a second, IL-6 can come from immune cells. That's the first thing we think of, but it can also be made by epithelial cells as well. Why that gets triggered in the milieu of the colon, but that could be a hard one to answer. But in the microbiome environment, as we advance to an adenoma to a cancer, there's been a number of associations with the and I've always wondered, when I first heard about this maybe 20 years ago, I was like, well, the real question is, what's the chicken egg? Is it because we have bacteria in our colon, and then the right thing happens, and then the cell gets irritated or whatever, or mutated, secondary mutated, and it grows? Or the genetics happen by whatever mechanism, and then as the growth happens, and so when these early reports came out, what is going on? Well, there's a lot of work done on this. I'm not at all, but there's been a lot of work done on this. Some of the answers are starting to be answered just now. When this was first described, just 2 days ago. So this is a summary from a few years ago, looking at the associations of certain bugs and the epidemiology. So there's some bugs here like streptococcus, and we know about the association of neoplasia with strep agalacticae and those types of things. But I'm talking about the ones that are associated directly with the cancers itself. So what has been shown over time, and I'm going to show you some of this data, that certain bacteria has been associated, not just with the epidemiology and evidence of microbial enrichment, now with some of the actual lesions by studies in the GI tract. And so things like early neoplasia, pulling back to produce oxygenic frag, They both make toxins that may have something to do with it as well. And Fusobacterium nucleatum and some of its subspecies have now been associated with late neoplasia, i.e. cancers and possibly even metastases. It can also make toxins as well. So this data, some of this data comes from, she's published several papers on this, some fascinating papers. So let me just walk this through with you briefly. So this is this study that was in Science a few years ago, looked at a group of FAP, lots of polyps, mice who are engineered to make polyps, right? So if you look at the FAP patients, she looked at particularly the enterotoxigenic B. frag and the PKS positive or Fuller-Back producing E. coli. And in this paper, looking at 25 FAP patients, 23 non-FAP patients, you can see that E. coli was a little more strongly associated with the polyps in the GI tract. So was the B. frag, and certainly the combination too was much higher than those who had controlled polyps or a single polyp. And of course, neither was the flip. So that was the patients. Then she took APC mice who make these polyps, right? And then she added the bugs. And if you add, enterotoxigenic B. frag, the mice live, 110 days and die. If you give the PS positive E. coli, they do the same. But we get the combination of both, the lifespan is just the bugs, same mice, everything, just a switch of the bugs. She looked at the colon inflammation. The back frag raised it a little bit. The coli here, but the combination of the two really raised the inflammation. Then she knockout mice. So they're not going to produce pro-inflammatory cytokine. Right? And so you can see that she colonized with both the E. coli and the B. frag. And you can see the type make lots of tumors expect. IL-17 knockout in the APC mice, not much tumors were made. So this tells you that the inflammation, or at least the pro-inflammatory, is driving those tumors. So the combination of those two polyps increase inflammation, faster tumor onset or survival, and appears to be pro-inflammatory cytokine dependent. Published actually a few years before that. This had to do with, again, she looked at the tumor cells, she called these biofilms. So she can do the biofilms on the normal, on the tumors itself. And the one thing that was tumors, particularly in the right colon, were these aqua colors, and these were all why am I blanking on the? Let me go back one. Fusobacterium nuclei. I had a brain infarct there for a second. The Fusobacterium, and you can see on this diagram, the Fusobacterium, particularly that makeup, look where all these tumors are from multiple people. They were largely in their colon. Okay. This Fusobacterium nuclei, I know there's some experts, party experts in the room. It's a bacteria that's largely associated with oral mouth. Why is this thing in the colon? Okay, it's an anaerobic bug. So the right colon makes the most anaerobic piece of the bowel, and that's where she found it. So she found the majority is right-sided. The ones that were negative was associated with normal mucosa, not the tumor. And if you had this, the risk of cancer was 5-fold higher if you had this Fusobacterium microbiome. Now, I also mentioned on the right side, that's where all our MSI high tumors Although I'm not going to talk about it today, that it's associated with both MSI high and EMAS positive tumors. Okay, but I'm not going to show. In this paper, this was all driven again by biofilm. So fascinating. If you look at those with versus no biofilm, these are the human tumors. The biofilm had way higher levels of IL-6 and the epithelium and the environment. And remember, IL-6 activates the JAK-STAT kinase STAT3, and activation was there, suggesting this was clearly down the IL-6 signal. The other thing too is that the crypts. So if whether you had colon cancer or not, film. So this, she can show this is staining for proliferation. The brown stains proliferation of these crypts. Normally the proliferation is way down at the base of the crypt, you know, where the LGR5 cells are. But you can see film, the crypt growth and proliferation is throughout the entire crypt. These are normal crypts. These aren't even adenomatous crypts at all. So that biofilm had a relationship with stimulated proliferation. Okay, so I thought that was pretty fast. The real fascinating one, this that a postdoc did in over at the Broad Institute, who now has their own lab. This is fascinating. So again, trying to get to the genetic egg study. What this script is, they had paired primary tumors in the same tumor that had metastasis. And they stained using RNA hybridization techniques. So here's a primary tumor, and you can see this pink on here. That's staining the fusobacterium. Here's a blow up. You can see the bacterium intimately associated with this malignant, area here. Okay, now down below, the liver metastasis in the same patient, and they're finding fusobacterium in the liver metastasis. Same thing in this primary, and here's the secondary. So the question is, okay, you got fusobacterium in his colon. Is the fusobacterium, two things can happen. You seed the tumor to the distant site, and then it gets reseeded by the bacteria. Or go with the tumor as it metastasizes. He thinks, this group thinks it's the latter. It goes with the tumor. So what they did is they took a human xenograft. So this is an actual human tumor, and they did serum passage in the mouse. And they did quantitative PCR and culture. Now, for 4 serial passages, they could culture it, meaning they put it in, they pass it, and so they kept detecting and could regrow the fusobacterium. They could detect it after 8 passages by PCR. So he believes it sticks with the tumor. Okay. This is also data from the TGA set showing the elevation of the fusobacterium. Now this was, I'm sorry, go ahead.
What's the
No, they're not using the tumor.
Yeah.
I'm sorry, they're injecting it usually in the flank, and they reculture it and take it to the flank, serial passages. Yeah, so they're not using the tumor. No, sorry, my bad. The fascinating thing is this, if that wasn't fascinating enough. So, fusobacterium, I told you, is an oral anaerobic bacterium. I just saw it because it's an anaerobic bacterium. And so you can take, these are HT29 cells, they're human colon all the time. Metronidazole does nothing to them because there's no fusobacterium. But you take the human xenograft that have fusobacterium, you get a shrinkage in the tumor. That tells you that fusobacterium is doing something to the proliferation, or at least success of that tumor. So he was able to shrink this in the mice by 30%, just an antibiotic. Okay, and that's also shown here. You can see the nitrazone. Okay. Now you may say, well, maybe that's the truth, but it's a pretty good study, and you keep publishing in Science. So I believe something there, I guess. So this is another group that had nothing to do with that study at all, and they did a totally just an empiric diet study. This is just daily, no mice on this study. And this group, they study is well, yes, the health professional study, and they were looking at association of bacteria in the microbiome status. And here's the fascinating thing: just lined up perfect. They found that the outcome. So proximal, distal, and rectal. That's how they broke it up. So they looked at, was it negative or positive for two? So the only ones that, so no statistical difference in rectal cancer, no statistical difference in distal, but only in the proximal, we'd expect diffusal, did they see a significant difference in those for outcome. So again, it lines up with the mouse data that I just showed you, as well as the diet data and the data, even the data that Cynthia Shear showed. So there's 3 or 4 different approaches to this. So just to summarize, and I'll close, the environment plays a major role in cancer formation, progression, metastases. Colon cancer, we have access to it, we can sample it, we can do a lot of things with it. And then, of course, understand this could change, diet intervention. I told you about the PI3 kinase and aspirin not used. We do microbiome intervention at some point, or inflammation prevention or modification for that. So some of the things I think about is, how does the environment influence differential genetic susceptibilities, like the PI3 kinase? So you can discover things, and then maybe the drugs we use or the chemopreventive agents we use can affect that. How does the environment contribute to disease formation? I showed you 2 examples from Cynthia Sears that really drove polyps in patients and in the mouse, as well as here associated with metastases. And then how does cellular pathways, how does the environment modify cellular pathways? How do we be susceptible so we prevent exposure? I mean, like I said, we have 8 billion people in the world. Are we to the point where we sequence everyone? Not quite yet. Certain key things that, hey, just like, could I predict that you could get hypertension at age 50? Or things change your diet. I can say, hey, your odds of getting a colon cancer are X, think about these things, And then also with disease. And then another thing, and I know there's some expertise in the room here about early onset, and I have some theories about that. But the environment probably plays a role in that as well. So I'll stop there and
I have a question about anti-PD-1 therapy and different types of colorectal cancer. So you informed us that when you have those frame shift mutations and those neoantigens, it can be pretty effective. But when I think about the classical pathway of like the APC, the KRAS to the P53, and tumors that develop like that don't necessarily have mismatch repair deficiencies, would anti-PD-1 therapy be not effective? Or
that's correct. So I'll tell you, it's not 100% explicit. So if you analyze all these tumors, and again, Jeremy, a lot of this, just even in the last few years, the majority of PD-1 responsive are MSI high. Okay. There is a minority of microsatellite stable that, for some reason, express PD-1 and anti-PD-1 and are responsive, but that's a minority, not the majority. It's largely driven by this concept of frame shift mutation. So there's probably something in the handful of MSS that may do it, or maybe it's completely independent of frame shift mutation. So it's not exclusive, but it's the majority. But even amongst MSI, we'll say the responsiveness is about 60 to 70%, because there's some immune escape mechanisms and other things. So when you look at some of these subsequent papers, and even the ones that you would predict to respond, it's still not 100%. But it's the mechanism for the PD-1 expression in the microsatellite stable happens to be probably something different than the frame shift immunogenic neopeptide mechanism.
We're a little, I suggest we do is close so the hepatologist can leave. And people that are interested can come up and talk to John. And I don't know if there's questions in the chat. Amy can moderate that. So thank you for, forgot when I introduced him at Link, I forgot to mention he's president of the H. Probably a big deal.
And so, here's the back.